and welcome to the Southern IPM Hour. Today is going to be really interesting, I think. It's on an ARDP project called Keep It Simple, so I'm excited to learn what that means. A sweet potato weevil integrated management plan for environmental and economic sustainability is the entire project title. And it is presented in partnership with the Southern IPM Center, where we present research issues and programs in integrated pest management from the Southern region through this webinar series, which again is the Southern IPM Hour. I'm Kayla Watson, the Communication Director of the Southern IPM Center, and we are housed at NC State and UGA with a mission to coordinate IPM across the entire region. We are also one of 4 regional IPM centers supported by the USDA NIFA Crop Protection and Pest Management Regional Coordination Program. And as I mentioned earlier, this project is an ARDP or Applied Research and Development Project, a part of the Crop Protection and Pest Management Program area at USDA NIFA. And we always want to let you know that this talk will be recorded and that a link will be available after the webinar today. If you have questions for the speaker as we go throughout the talk, you can put those questions at the Q&A box. That's a little bit different than the chat box, but it's still at the bottom of your Zoom screen there. And we'll answer those questions at the end. So if you have a question as we go throughout and you don't want to forget, you can put that in at any time. Our speaker today, though, is Jeff Davis from the LSU Ag Center. Welcome, Jeff, and we're excited to hear your talk.
Thank you, Kayla, and thank you for everyone who's on today. Good morning or good afternoon, wherever you may be. And yes, the title is Keep It Simple, a sweet potato weevil integrated management plan for environmental and economic sustainability. Let's see, is it going to advance my slide? It may take a minute. Just to let you guys know, this research was supported in part by USDA, NIFA, under the Crop Protection and Pest Management Program Award, under my Hatch Project accession, and under the Louisiana Ag Experimental Station. And of course, the findings and conclusions, as you guys have seen this before, are preliminary and should not be construed to represent any agency determination or policy. So here's just a quick overview. I'm going to talk a little bit about sweet potato in general and the importance of sweet potato weevil. We're going to talk about what we tried to do in shifting the paradigm, the team that we had, the trials and tribulations of research, and what we accomplished. As you know, sweet potato is vegetatively propagated, or just to remind you, and it's prone to accumulation of pests and diseases, and it is a very hands-on crop. These are some various pictures showing in the top left corner. Here's my group getting ready to bed sweet potatoes. We typically in Louisiana are bedding sweet potatoes in March. You have to lay those out. You then cover those in black plastic. In April, you remove the black plastic. The slips or the storage roots sprout. Those sprouts then form slips that are in the lower left corner where you cut those and then get ready to plant those in the top right corner. This is, like I said, very hands-on. So here's my group planting our sweet potatoes. And then even when you harvest, you dig those sweet potatoes, they go up onto usually a platform where you have people sorting those if you're a grower, throwing out ones that do not meet the quality standards typically for sweet potato. Some of those in importance are these soil-dwelling insects because the storage roots are grown underground. And you can have various blemishes and things that would probably discount your sweet potato caused by different insects with banded cucumber beetles, showing this nice scalloping, white grubs, wireworms, and sweet potato weevil. Now, sweet potato weevil is an invasive species. In Louisiana, it was brought into the state in the late 1800s. It is throughout a lot of the sweet potato growing area throughout the world. Stylus formicarius elegantilis. It's a beautiful sweet potato weevil, showing, but very small, that can cause a lot of damage with its ability to feed in and on those storage roots. In Louisiana, we do have different zones that are maintained by the Louisiana Department of Ag and Forestry. Those zones are green tag and pink tag. And these are very important, so I'm going to spend a little bit of time explaining what those mean. And of course, in Louisiana, instead of counties, we have parishes. Now, in green tag zones, there are no weevils found there. Those are supposed to be weevil free. And only seed from the previous year's green tag fields can be planted in those green tag zones. So let me go back. Those are in the upper corner of the state, upper northeast corner of the state are the green tag zones. But we still have a lot of sweet potato grown in the Avoyelles, Rapides parishes, Evangeline in the center of the state, as well as Acadia, Lafayette, and Point Capee and St. Martin areas. There's still sweet potatoes grown. So there's grown in both the pink tag and green tag zones. So once again, green tag, you can only plant storage roots from green tag zones. And no planting is allowed within 100 yards if a sweet potato weevil was trapped there the previous year. Any sweet potatoes found in that area have to be then destroyed. If they're found in that field within 300 yards, they will be declared a pink tag. And that will last for 3 years and have to be, once again, monitored. And so there's this continuous monitoring for sweet potato weevils in the state and in these areas. That's done with pheromone traps and is run by Louisiana Department of Ag and Forestry once again. In the pink tag zone, when you plant those beds, as I showed you in the previous one, you have to start spraying at 10-day intervals until those beds are destroyed. And by law, they have to be destroyed by July 15th. In fields, once those slips are planted, fields then have to be sprayed every 10 days once transplants have been planted. And so typically sweet potato is sprayed from April to September every 10 days, or approximately about 18 applications per year to control sweet potato weevil. So very important, a lot of effort done with this. Now, once again, weevils are found in Louisiana. They're found in parts of Mississippi, South Carolina, Arkansas, and Alabama. They're not found in North Carolina or California, two of the biggest sweet potato growing areas. So the sweet potato weevil, very important for our area, especially for the pink and green tag zones. The weevil, the injury that they cause are by feeding on those harvestable tissues. You can see these are emergent holes coming out of the storage root itself, and it induces these furanoterpenoids. Okay? And what's actually feeding within the storage root are these weevil larvae. They burrow through. An egg is first laid by a female. Now, a female sweet potato weevil has to actually feed on the storage root itself. She then turns around, oviposits one egg in that feeding hole, and then she covers it with her frass or her insect poop. Okay? That egg then hatches, then burrows through that storage root and can cause these inductions of those furanoterpenoids. As that larvae goes through its stages, it then pupates and then emerges as adults, where the whole process starts over again. Now, those terpenoids give this reduced palatability, off flavor, or off taste. And I'm using an example where this is a storage root that's been fed through. And here's that classic example in science where they fed blue jays different larvae of insects that had been fed on, say, plants that they acquired some different insect secondary plant compounds, causing the bird to regurgitate. Essentially, what's going to be the same is it gives this like off flavor, off taste, and the presence of a single weevil larvae is thought to render the entire root unacceptable for human consumption. So zero tolerance for weevil damage, very important. For animals, if fed these storage roots, so there is some areas where sweet potato storage roots are fed to, say, cattle, especially in the continent of Africa. These terpenoids can actually cause some real problems. They're toxic to those animals and can lead to death. So it's important not only within the nation, but within the world as well. So our idea was to shift the paradigm that's been going on, is to try to reinvestigate if these weevils truly do have an impact, this furanoterpenoid induction, and if it does implicate in some palatability. So what we wanted to do was look at amount and spatial extent of that terpenoid induction. In other words, can we cut part of the storage root and still be free of terpenoids? and then to conduct sensory assays with consumers to determine if the current zero threshold is warranted. So we wanted to fear no weevil, as those little signs are shown below. The second part of our project was really to reduce the amount of insecticide being used, to combine it with host plant resistance, and to use entomopathogens. And once again, to develop non-insecticidal tactics for weevil management, to reduce the environmental burden of the current program, and to see if we can suppress at least weevil populations equal to what's contained with insecticides. With 18 times being sprayed a year, eventually those insecticides are going to run out. We probably will see resistance building at some point, and we need to be able to protect our producers of sweet potato. So why look at host plant resistance? Well, I'm an integrated pest management guy. I use host plant resistance all the time, but just to remind us, it's compatible with the other controls of IPM. So it can be used with cultural controls. It can be used with biological control, which we're going to talk about. It can be used with chemical control. It's persistent. Once it's released, it's out there. It's specific to the specific pests that you're trying to control. So you're not going to get that broad spectrum effect. You're not going to be killing off the friendly insects, our natural enemies. It's environmentally friendly, and it's easy to adopt, right? If it's out there for the growers, if it's in their cultivars, and those cultivars are high yielding, it's important that they have this host plant resistance. Why use entomopathic fungi? Well, once again, these are naturally occurring fungi that some of these possess insecticidal properties, and many of these are actually commercially produced. We have various Bavaria, Bassiana, Metarrhizium strains that are available, and they work very good and very specifically against our insects. So we wanted to be able to, once again, combine host plant resistance, combine biological control, combine these integrated tools and techniques to be able to provide growing conditions that we didn't use necessarily insecticides. And what were we going to do? Well, you know, we do what the LSU Ag Center does best. We do our research, our extension, and teaching. We had a multidisciplinary teamwork approach. We actually had a teamwork with private industry, Ag Biome, which was helping us out with our different entomopathogens. We did on-farm and on-station research and extension. We worked with various growers, both in pink tag and green tag zones, to allow us to do this work on their fields. This project trained our next generation of scientists, and it helped the producers of Louisiana grow more food with less. That's the whole idea. That's what I do, and that's why I love my job. Here's our multidisciplinary team. Entomologists, myself, Dr. Mike Stout, Nupur Sakar was the postdoc on the team. Tara Smith is our sweet potato extension person. She's now the vice president or associate vice president for extension, but she still works in sweet potato. As I said, Chad Kaiser was with the Ag Biome, so a private company. Chris Clark is our plant pathologist. Don Labonte is our sweet potato breeder. Zijin Liu is our plant chemist, and Dr. Watoon is our food and nutrient scientist. So he was going to do the food panels once we did our tests. But of course, all science doesn't go well. There are various trials and tribulations. And one of the first things was that when we got this project back in 2020 and started to conduct it in 2021, COVID happened, and that really reduced our ability to hire people to do research. At the same time, there was a building fire in my laboratory that pretty much shut down the building for another 7 months while they fixed all the problems that happened because of that. Ipomea marrone, the main furanoterpenoid, could not be purchased as a standard, so we thought it could. When we went to do so, there was only 2 companies that currently made it, so even if Fisher Scientific says it's available, don't believe them, it might not be available. One of the companies was a prohibited source, so we could not purchase from them, and the other one was cost prohibitive. It was going to cost us hundreds of thousands of dollars, and that was not available in the grant for that. So necessity is the mother of an invention, of course, as they say. And so we went around, if we couldn't purchase it, we had to make it ourselves. So the furanoterpenoid was going to be very important for us to understand as a standard for our different chemistries that we were going to be running, to be able to know, if those insects, if the plant pathogens were inducing this furanoterpenoid. So when we infested storage roots under sterile conditions, and then we had help from the Department of Chemistry because we couldn't do it all on our own. And using LC-MS-MS, we detected our Ipomoea marrone and one of the other furanoterpenoids. We extracted those using the HPLC packed columns. We then had to identify and confirm all this using techniques that I'm not always comfortable on trying to understand or be able to talk about. So it's better for a chemist to talk about that. Needless to say, I do list the techniques. We did publish this. And we were able to isolate ultra-pure Ipomoea marrone for quantification of the accumulation in our storage roots. Of course, all this took 1.5 years to accomplish. This was a 2-year project where we did get a no-cost extension for an additional year, but we're even still to this day still doing research using that Ipomoea marrone. So our first objective, we were going to reinvestigate weevil and plant pathogen impacts on furanoterpenoid induction and implications in palatability. One of the first things we did is that we infested storage roots. So we took storage roots, we infested one part with sweet potato weevils. We allowed the females to oviposit, lay their eggs. We then took those storage roots, put them in containers separately, eggs hatched, and we looked at larval movement throughout the storage root from one end to the other, as you can see in this graph. Up on top, you can see that the different 1 centimeter slices that we took throughout the storage root to try to then identify Ipomoea marrone accumulation. And here's our concentrations in parts per million on the y-axis, on the x-axis, distance from infestation lines. And you can see that the closer you were to where the weevils had either been burrowing or in like 4 centimeter where they still were detectable just outside of where those weevils had been burrowing as larvae, you can see that there were pretty high levels, and then those quickly dissipated the farther out you got from that storage room. In terms of this Ipomoea marrone on weevil oviposition itself, in red, that same graph that I showed you on the previous, here on the y-axis, we have oviposition. So it's important to see that if those Ipomoea marrone levels were very high in the storage root, it actually deterred other female sweet potato weevils from ovipositing on there. So once again, this is an impact of the fact that a weevil would be feeding, causing these terpenoids, and then reducing further colonization by the sweet potato weevil. So very interesting. Could we use that in the field? That's something we're still working on. So in conclusion from that, we did not accomplish the plant pathogen impacts on Ipomoea induction or implications in palatability because we just never got to that point and weren't ever able to do it because we didn't have the Ipomoea marrone. Once we had it, we were able to at least do the parts with the insects. We're still continuing to work on that. But once again, you know, sometimes what's a good example is that though we give out these objectives, sometimes it's not always able to accomplish those in the time frame that we had, even with the 1-year no cost extension. But we did see that weevils do induce and accumulate Ipomoea marrone in the storage roots. These females can identify those concentrations, and maybe this is a gustatory response because, like I said before, the female has to physically feed on those storage roots. She then takes in part of that sweet potato, determines if it's an appropriate host before she then goes and oviposits. And so this is part of that good mother strategy. In other words, she's choosing where her progeny are best going to be at. And what's seen is that they can identify these concentrations, probably seeing that already these storage roots are infested and that it would be maybe a detriment to her progeny to be able to lay those on that storage root. High concentrations reduce that oviposition, and that parts of the storage root are terpenoid free after injury. And that's very important so that some of these storage roots that are culled, that are thrown away, maybe they would still be usable, and at least parts of those for, say, yams that might be put into canned yams and things like that, or maybe still are okay for animal feed. Objective 2, we wanted to reduce the amount of insecticide used by combining host plant resistance and entomopathogens. And the first thing we did is we looked at, do the varieties that we had from our breeder, Dr. Don Labonte, do they differ in response to sweet potato weevil? And in other words, we were looking for host plant resistance to the weevil itself. Now we picked various varieties to test. Beauregard is the kind of the industry standard. It's been around the longest. It's still one of the largest acreages grown. It's a very typical sweet potato in the fact in the United States that it has orange flesh, kind of that orange skin. Variety Vermilion was released in 2021. Here is a sweet potato that has more of a purple kind of skin and once again that orange flesh. And we didn't know anything about this variety at the time. Variety Murasaki, once again, purple skin, but it's a white flesh. This is more typical of, say, a sweet potato that would be grown in Africa, that would be grown in Asia. And it's more of a used for starches than, say, that sweetness that's found in there. Murasaki was already kind of known. My lab had done some work showing that it's very resistant to sweet potato weevils. In the fact for emergence and other things, we've published that work. And it's just a good sweet potato in itself to eat. It's high. All of these varieties will be high yielding, fit into growers, meet grower standards and things like that. So that's the varieties we were looking at. Now, variety Orleans, once again, more of a susceptible variety. We already knew that, so we had 2 susceptible varieties, 1 resistant variety, and this new variety that growers were planting. And we picked the varieties that were the most numerously grown in Louisiana. And we chose those based on not only our agents who work with our sweet potato growers, but our sweet potato growers themselves, the varieties they use. And so the work that we did, we looked at using no-choice assays, looking at sweet potato weevil emergence. On the y-axis, you see the net mean adult emergence per root. What we would do is we would infest those with 6 females, then take them off, have the storage roots. It takes 30 days for the weevils to start to go through their whole life cycle from egg to adult. They then start emerging, and we look at emergence throughout those. And with Beauregard, we saw those were very susceptible. We saw lots of weevils emerge from those. Murasaki, we already knew there were going to be less emerging from those. And lo and behold, Vermilion, still more of a orange flesh, so it meets even a better industry standard. And here we have resistance equivalent to Murasaki. So that was really great. And it's one of the key examples of our work that's really been useful for our growers. If we did some feeding weevil feeding and oviposition assays, once again, also in the laboratory, and you can see on the left side of your screen, this graph showing feeding preference. Here, amazingly enough, the one that the weevils emerged out of more often, actually they chose, when given a choice, they fed less on Beauregard than they did, say, Orleans. Murasaki, once again, less weevils emerge out of Murasaki, but actually has a higher feeding preference. We didn't know that before. Now we understood that better. And if we looked at the oviposition, this is on the right-hand side on the graph, here we see that, once again, Beauregard is chosen less often than Murasaki, are more resistant. So can we say Murasaki is resistant in terms of oviposition or feeding preference? No. But if we talk about it in terms of emergence and looking at the weevils that do emerge from Murasaki, yes. Orleans, Vermilion, all much lower. Murasaki, by far, the weevils prefer to feed on. The eggs are laid, but once those eggs hatch and the larvae go into those storage roots, they do not survive. And only about half of the eggs that are laid actually emerge out of Murasaki. So very interesting. Can entomopathogens be used in our sweet potato weevil? So we had to go back once again, do some studies looking at, could we use those? And we chose various products that we could find online. We did make sure that we used an industry standard. So we used Brigade. This is typically used by our sweet potato growers. This is bifenthrin. We compared it to Metmaster and to Bottega ES. Both of these we could purchase. And then we had an untreated check. So the 2 entomopathogens, Metmaster, which is the Metarrhizium, Bottega, which is the Bavaria, that we used at different ounces per acre. In the laboratory, we do laboratory chip assays. We were looking at mortality. You can see with the Brigade, with the industry standard, After 3 days, we were already at almost 100% mortality when we used a chip, a sweet potato chip that had been soaked in the pesticide and given weevils to feed on. Bottega did very well. We got close to 75% mortality after 14 days. To me, that's a win. Any product that gives close to 80% control is a positive. Metmaster did no better than our untreated check, and it just did not perform very well in the laboratory. We did continue to study it throughout our work. The next, we did these little laboratory terrarium assays. Here, my postdoc Nupur came up with the idea of being able to grow some slips in these like little jars, be able to spray the leaf tissue, right? Because we were looking at both feeding on the storage roots important, but the adults feed and are moving through that foliage. And so it's important to see if they can still, if foliar applications can control the weevils as well. And once again, days after treatment, mean proportion of mortality. Brigade, once again, worked very well to control weevils. Bottega, a lot better when given and controlled the humidity. So the humidity was higher in these jars. And that's something to think about when using entomopathogens. They have to have high humidity. Usually not a problem in Louisiana. We have very high humidity on a regular basis. And then Metmaster, untreated check, not any better. So once again, here we have products that are working, able to do these in the foliar, working on the storage roots as well in the laboratory. We then move to the greenhouse because that's important to be able to study that as well. Looking at different varieties, once again, seeing if there was a combination between variety and the effect that we were going to see with these different products. Looking at mean proportion, we would grow the storage roots out. We would have these and applying this to the foliage in greenhouses. Once again, we had to use cages because we did not want to release weevils out into the nature. Even though it's normally found in Louisiana, we have other research going on in the greenhouse and we did not want to affect that. So you can see we have these little cages that we put them in. Once again, Brigade worked the best. Bottega working very well with both Beauregard, Murasaki, Orleans. Look at Vermilion. Vermilion was one of the varieties we said was resistant. Here we start seeing where there might be some antagonistic effect between the entomopathogen and the weevils itself, right? Because we did not see as high of mortality in Vermilion. It was something we looked at and continued to study even to this day. Metmaster, untreated check. We did have some mortality occurring in the greenhouse, even in our untreated check, which was just water when we sprayed those. I forgot to mention we sprayed these using a mist sprayer or a CO2 backpack sprayer like we use typically in the field when we put out our trials. Greenhouse residuals, because we weren't just looking at the impact, allowing them to feed on the foliage itself, but we had sufficient foliage to take at different days after treatment, bring it back to the to the laboratory, feed it to the weevils in the laboratory and look at the effectiveness of these products over time. Now, what's interesting in the greenhouse, of course, we're not getting water, so we're not getting rainfall, but you are getting UV that's going to be affecting, right? Even in a greenhouse. So you're getting that sunlight that can break down some of these particles. And we can see that Brigade working very well. But once again, here's Bottega, where if you see that relative humidity, and we tried to keep it high in the greenhouse, but under certain conditions in Louisiana, that sunlight's pretty high and we can't keep the temperatures always regulated, even in our greenhouses, or the humidity. And we started to see some effects of the UV light on Bottega on our Bavaria. Our Metmaster didn't work at all, no better than our untreated check. Got to make sure that I'm on time. Looking good? All right. So will the entomopathogens, of course, we did all this work in the greenhouse, in the laboratory. Now let's take it to the field, working with our growers, working on our research fields. We did this multiple times over the 3 years that we did this product. Could they reduce damage to storage roots in the field? From our field foliar assays, we did residual assays again. We took and we sprayed these on the foliage on plants that prior to canopy closure. We then sprayed them at canopy closure and 2 more times. So a total of 4 times throughout the field, throughout the year, so that we could make sure that we could try to build up that Bavaria in the field. But even from our foliar assays in the field, we've seen once again that Bottega breaks down very quickly in the field. Didn't even work very well at all with only 30% control, even after 0 days after treatment. That was actually within 2 hours. We went back, took leaves that were dried and tested those, and we saw only about 30% mortality. And we can see Brigade even breaks down very quickly. At 7 days, you're still getting maybe 60% control, 70% control. At 14 days, it's very ineffective at that point. From our field trial to look at below ground herbivory, we harvested the storage roots, brought those back. We assessed those, not only for our sweet potato weevil, but for batted cucumber damage, our white grubs, and our wireworm damage. From the previous work, I forgot to say, all I'm showing here is with the insecticides. With the host plants, we saw no differences between the different varieties. So vermilion worked just as well as Beauregard, or was just susceptible. So the combinations, it was just the pesticide by itself. Here, for storage root injury, when we assess those, if we looked at banded cucumber beetles, we see that Beauregard very high. We see a lot of feeding scars in ours if we don't control with banded cucumber weevil. But with our Murasaki and vermilion in our untreated checks, we see that vermilion is holding up very well, almost as resistant and equal to resistant to Murasaki. So here you have this variety, high yielding, resistant to not only our weevils, but also in the field, resistant to banded cucumber beetles, resistant to our Phylophaga. So those are going to be your white grubs. Looking at meat and feeding scars again, vermilion showing very good resistance. And even our, when looking at our melanatus, our different click beetles, our wireworms, we see that vermilion, once again, showing just as resistant as Murasaki. So Beauregard always susceptible, vermilion holding up very well against these other soil-dwelling insects. So just for our objective 2 conclusions, vermilion we were able to show is resistant to sweet potato weevil, cucumber beetles, white grubs, and wireworms. Awesome variety. I hope it picks up in use by our growers. It's very widely used in California right now. I hope it starts being used in our area more, because it does have this resistance to our storage, or our storage root pests in the ground. It's not a preferred or feeding for oviposition or for feeding. An adult emergence from the variety is much reduced for our sweet potato weevil. So that's a positive from the things that we were working on. In terms of a greenhouse or laboratory setting, Bottega worked very well. But when we got to the field, it failed. Maybe it failed because of UV and heat inactivation. Maybe it failed because we were applying a foliar. We did not get a chance to do our in-furrow applications. We could also try soaking the storage roots and putting those in. These are, or our slips prior to planting. We're trying to do that, going to starting this year. But mainly at this point, from a foliar application, even doing 4 applications did not provide that protection in the field. And we were not able to reduce the amount of insecticide used by combining host plant resistance and entomopathogens. So unfortunately, though it was one of our goals, it looks like, though we did find new host plant resistance, really insecticide use is the main control. And it's not only that, it's in our pink tag zones, you have to spray. This is by law. And better entomopathogen formulations may be needed. Really, that UV protection is probably going to be the main problem. In Louisiana, like I said, it's not the humidity. We have lots of it. So overall, this project trained 1 master student, trained 1 postdoctoral researcher. We were able to identify host plant resistance to sweet potato weevil, cucumber beetles, white grubs, wireworms, and an orange flesh high-yielding variety. This is very important. This has been one of the goals of the breeding program under Dr. Labonte for years. We've reached that now. This variety, though, is very specific. It likes certain conditions. For instance, the 1 year we planted it in 2021, or in 2022, we had a lot of rainfall. Vermilion does not do well, yield well, under high rainfall conditions. Under real drought conditions, it seemed to do very well. We established new collaborations within the Department of Chemistry. Not only that, we were working together as a team to be able to do all this research, and we were able to at least get milligrams of ipomeamarone for further research. And so this will lead us to being able to apply for new grants, continuing to fund this project. We generated outputs in the form of four peer-reviewed journal articles. 3 of those are in press right now. We had 8 research presentations at regional and national meetings, 3 extension and outreach field days, 1 extension video. Here's the link. I think Kayla said she was going to post it in the chat as well for people to look at. And with that, I just once again want to thank the support from USDA, from the Crop Protection and Pest Management Program, our Hatch Project, Ag Experiment Station from Louisiana. And to remind you, this is all preliminary, and some of this still hasn't been published yet, so please take it with a grain of salt. And thank you. And I probably a little early, but that's okay. Maybe we'll have lots of great questions. Here's my information, and you can feel free to contact me if you have any further questions. Thank you.
Thank you so much, Jeff. That was awesome. And I'm going to go first to Hannah. Hannah's had her hand up for a very long time, but I did promise to save those questions to the end. So Hannah, I'm going to allow you to talk, and you should be able to unmute and ask your question to Jeff if you'd like.
Oh, sorry, that was my mistake. I'm sorry.
That's fine. I felt really bad the whole time, but that's okay.
Oh, no, sorry. I had someone interrupt me halfway through and I accidentally clicked. My apologies.
No problem. Just wanted to make sure we got this questions answered if you had one. So just as a reminder, you can do like Hannah, and you can use the raise hand feature to and I can unmute you to ask a question if you'd like, or you can just put your questions in the Q&A box. And we do have one question there now, and it says, are you breeding the sweet potato weevils for these experiments, or do you collect them from the field?
Yeah, great question. So we actually have a colony that we keep in the laboratory. We go back out to the field every year, get new collections so that this colony is continuously going. We rear them on Beauregard itself, so but we do add in fresh weevils from the field. We've been doing that since about 2009 was the last time that collection was made to continue this research. So yes, we have a process to continually rear our sweet potato weevils. You just need lots of sweet potato storage roots.
Gotcha. Thank you. Another question that we have is from Roger, and Roger says, are you looking into testing formulations of biocontrols that may include UV protectants?
Yeah, that'd be great. So if you know of anyone, Roger, who may have some of those, we're always looking for new products that we can use. Yeah, that would be good. We also had a project with AgBiome separately from a different grant, and we're looking at one time hoping to be able to do that. Currently, that project's finished, and we're always looking for more supporters. So yeah, if anyone knows of anyone, I put it out there. Come see me. Here's my contact information. We can work together in trying to do that. So yeah, I think entomopathogens, again, are still one of the important parts of agriculture. I think they're underutilized on large crop acreage. You know, from me, I usually work in soybean, corn, cotton, things like that. And more of those need to be put out there because we need products that are equivalent. Our growers will tell us we need products that are at least equivalent to what the synthetic chemistries that we have currently. So once again, that's important to know and are as effective.
Always good things to consider. So Jeff, I'm not seeing any more questions coming in now. But I wondered, you know, you did this whole big project for years, and I know you ran into all of these problems and everything, but what do you think was the most interesting finding that you had from this project?
Yeah, that's a great question. Okay, from a scientist standpoint, I think just seeing a variety like vermilion being resistant that has still that orange flesh meets the U.S. standards for a high yielding variety is probably the coolest thing. I know maybe you all don't think that. That's okay. I think it's pretty cool because it's having been working in integrated pest management for years, working in potatoes, sweet potatoes, soybean, finding a breeder who wants to work with an entomologist can sometimes be difficult because we are going to destroy your products, your varieties. The whole purpose is that we're testing to find that resistance, and to be working with Dr. Labonte and getting to that point where we have a variety that's perfectly acceptable is great, because often we get early varieties or advanced breeding materials that don't quite size up, they don't give the yield, and they don't fit our grower needs, and this one does, and to me that's great. On a non-research point, just working with all those people was difficult, but it's always a lot of fun. And the more growers that are involved, just seeing the smiles on their faces, that's what keeps me going. When I get out to that field and talk to them, I spend too much time in the laboratory often, but that's part of my job. But getting out and working with farmers, that's my fun time, because that's the best. And that's when you find out if your research is really being accepted or not.
I'm just learning so much just from hearing you talk about your research. It's amazing. So just my final question just for today is, as people are going throughout their day or thinking about sweet potato weevil or your project, what is one or two like main takeaways that you would like for them to have as they learn more about your project?
Sure. It's okay to eat a storage root that has some damage to it. That's always one thing, right? So, one of the things I like to think about is that food doesn't always have to be pristine. Yes, I like going to the grocery store and finding my shiny apple, but I also like going to and picking my own apples that have a little bit of damage to them, because sometimes, if there's a little fungus on there, maybe it's already converting those complex sugars into those nice simple sugars that I want to eat and be able to get my own gustatory response. And if you know me, I like to eat a lot. So I think in terms of my research, it's important to understand that, a little bit of insect damage does not mean it should be thrown away, and that a lot of work has to be done as a team or we're not going to get there. And one final thing, don't be afraid to change the paradigm. If just because it's always been said that one weevil causes the complete storage root to be thrown away, don't believe it, test it yourself and see if you can do it.
All right, Jeff. Well, thank you again so much for everything that you've told us today. Oh, we actually do have one more question if you're up for it.
Yeah, sure, go ahead. I'm fine.
Can you talk a little bit about the biology of the weevil in terms of how it would come in contact with insecticides when they're applied in the field? Is the weevil spending significant time above ground?
Yeah, so that's part of the thing. This is a cryptic species. The female has to get down to the storage root, right, to lay its eggs. We've worked at looking at, if we take it into the lab and we cover it with even a thin layer of sand, can this weevil dig into the storage root? No. The weevil actually has to have cracks in the ground surface to get to the storage root. Okay? It also can feed on the little sprouts that are coming up out of the ground, can kind of push the soil, and it can get to it. So one good cultural technique that our growers do anyway is when they hip up their rows and throw more soil up on top of those rows to cover. Now, mind you, you can't do that once those vines start vining out, but you can do that early season. So yeah, biology-wise, the females, the males will spend a lot of time in the foliage, but they tend to be more nocturnal. They tend to be spending time, if they can get into the ground via cracks, they will. But they cannot physically themselves, at least in the laboratory and greenhouse work we've done, dig into the soil. Even potting soil, they can't dig into. They got to have a crack. If we physically create like just a little hole, boom, down they go in. In terms of biology, yes, they'll get exposed in the foliage. They are hanging out in there. You'll find them underneath, but they're moving around. They're moving around. They'll go looking for any morning glory they can find. So if you have a bunch of weedy species out there, weedy morning glories, they're going to be out in these fence rows. They're going to be hanging out around in these non-crop areas and then move into your crop as soon as they get a chance to. So I hope that answers some of your biology question.
Just a follow-up to that. When are beetles most active? When are the beetles most active?
They're pretty much active. I've seen them active. We'll take pheromones out to the field and check. I've seen them active March all the way through till December in Louisiana. You know, I mean, we can have 80 degrees in December and they're still moving around. They probably go through a little bit of a diapause, but it doesn't ever stand that long. You know, we bed in March and we start planting in May because May is yam spelled backwards. And that's typically what we're doing. And yeah, since it's May 1st, it's yam first. And I need to get out and start planting.
Let's see. Okay, we're getting more questions now, so that's good.
Of course, good. I broke the ice.
Yes. Let's see. Has spraying at night when the beetles are more active been experimented with? Have you tried that yet?
I haven't, but I know often our growers are going to be probably spraying at dusk because you know, just by winds, right? You don't want that high wind speed that seems to pick up. So our growers are probably spraying either early morning or at dusk, but I have not studied that myself. That's a great question. I'll add that to my long list of things that I need to do yet for this insect.
Then another question that we have is it understood how many generations occur in Louisiana?
Yeah, that's a great question. It takes about 30 days from egg to adult under typical Labrador, you know, so at about 25 degrees C, which is, you know, you're looking at, you know, 77 degrees Fahrenheit. My guess is that we have multiple generations occurring. As to the exact number, probably anywhere from March, April, May, June, July, August, September, October. I bet there's, you could probably get close to 8 generations a year.
Wow.
You know, they're going to be continuously, if there's weedy species Ipomea species that have storage roots, they're going to just be feeding and growing all the time.
That's a lot. Wow. Okay, so I think we made it to the end, Jeff.
Awesome.
But
Thank you all very much. Yep.
Yeah, and thank you so much for answering all those questions for us. And don't forget that he's got his email address right there for you. If you have any follow-up questions for him, I'm sure you'll be happy to answer those. And thanks just for being a good sport on answering all that questions. Just to let you know, there will be a quick pop-up survey after the webinar. It's just 5 questions, and that helps us guide the future of our webinars. So it's just a few questions about, you know, things that you learned and things that you would like to see for future webinars. So if you could answer those, that would be extremely helpful for us. We do have a couple of webinars coming up for the Southern IPM Hour in June. We're going to have a webinar on school IPM. And then in August, we're also going to have Southern IPM Center offerings. So anything from grants to our awards program and to a new travel grant that we're offering as well. So you can check that out in August, and you can see all of the upcoming Southern IPM webinars at southernipm.org. And don't forget that a link to the recording of today's seminar will be sent to your email and available on our website once we are able to get that up, probably about a week or so. So thanks again to Jeff at the LSU Ag Center, and have a great rest of your day.
Thanks, everyone.